Well, not all viruses are the same. They're different in type of genome, if they have or they don't have an envelope, or the nature of their self-surface proteins. They could have what kind of receptor they have, and also the type of life cycle it can enter. It can either go into the lysogenic cycle, the lytic cycle, or the productive cycle. And because these viruses are unique to one another in terms of their characteristics, like the ones I just mentioned, they can be classified based on them. And one good way of classifying viruses, or one way that we classify viruses, are based on its genome. And of course, we know that we have DNA, RNA, circular, whatnot, but we haven't discussed about it in detail. So you might be watching and you'd be like, what's this positive RNA virus and what's a negative RNA virus? So don't be worried about what that is, and because we're just about to find out what it is just now. So here is a virus that's going to infect a host cell. It infects it through endocytosis, and now it's going to be traveling deep into the cell. And now when it's deep into the cell, it will burst and make its genetic material available. And we find out that it's a positive RNA virus. If it's a positive RNA virus, just remember that this is the virus, this is the strand of the RNA virus that has the ability to produce the viral protein. It's the virus strand that actually allows the characteristics of the virus to be expressed. So first of all, we will have the RNA-dependent RNA polymerase that is firstly translated. This is very important because this is important to understand because the RNA-dependent RNA polymerase will help in the process of replicating for the viral genome. But you might be confused in what is this RNA-dependent RNA polymerase? What does this stand for? Let's break it down. Well, first of all, the RNA polymerase is just referring to the transcription of the RNA virus, right? So that's pretty understandable. Then we have the RNA-dependent. That this part means that if you think about it, this RNA polymerase is being translated from an RNA virus or the RNA strand. So it's RNA dependent because of that. Now that we have a RNA-dependent RNA polymerase, we will have replication. And replication of the viral strand happens in a 5' to the 3' end, obviously. That's the direction for transcription always. So now we have the RNA-dependent polymerase doing this thing. And as it's being transcribed, it's not going to transcribe a positive strand because we know that because every kind of genetic information has, or DNA has, or RNA, any kind of strand, nucleotide strand, has a complementary form when it's replicated. And that complementary form, in this case, will be the negative strand intermediate. Now, you might be able to connect this with the negative RNA virus, and indeed, this is pretty much a reference to that. The negative strand does not have the ability to produce the viral proteins. And it's only the positive strand that has it, so it's considered an intermediate. So in the sense that it has to be transcribed to make the positive RNA virus strand, which will be responsible for all the viral characteristics. Now let's go back out and zoom and quickly go over the positive RNA virus now that we understand that the positive RNA virus is responsible for the viral characteristics. So the positive RNA virus is required to encode the RNA-dependent polymerase, or the RNA-dependent RNA polymerase. And we saw that right over here, the entire process, that it does indeed encode for the RNA-dependent polymerase. And it doesn't need to carry one, and that's because it is the positive strand, and it has the ability to produce the viral proteins, and it's not the negative strand. Now, getting on to the negative strand. If it's a negative strand, it's the intermediate strand of the positive strand. And that means that it doesn't have the ability to produce the viral proteins. And the only way it can actually do that is by somehow, being transcribed to its positive RNA virus, which is its complementary. So because it's a negative, it doesn't have the ability to produce really anything. It's stupid, or not stupid, but it doesn't have any kind of purpose in the sense that if it's translated, what I'm trying to say, if it's translated, the polypeptide that emerges out of it will be nonsense because it doesn't make any sense. So it physically needs to carry an RNA-dependent RNA polymerase. And once the first negative RNA virus is transcribed to a positive, then that positive RNA virus can behave in a behaves in a very similar fashion, and then it has the ability to encode for more. So let's understand the negative RNA virus by a couple questions. Why do negative RNA viruses need to carry RNA-dependent RNA polymerase? Well, we've talked about it just now. The positive RNA strand is the coding strand. To replicate the first positive RNA strand, it physically requires a RNA-dependent RNA polymerase, and that makes sense. If a negative RNA strand is translated, will these polypeptides be viral protein products? No, it's complementary is useless polypeptides that are, if we were to synthesize a negative strand, it'll be useless polypeptides that are synthesized. Understand why is it called RNA-dependent RNA polymerase. We also talked about that, surprisingly. Well, these are my notes, but still. RNA polymerase used in viral replication are made by RNAs. Therefore, RNA-dependent RNA polymerase. So whenever you see RNA-dependent, just understand that the RNA polymerase is being derived from the RNA itself. So therefore, it's dependent on the RNA. Now let's talk about retroviruses. Now retrovirus, and here's an example of a positive RNA retrovirus. So the end goal of a retrovirus is lysogeny, and lysogeny just refers to the lysogenic cycle, where it wants to incorporate its genes with the host cell's genes. And if it's a DNA, then of course it has to somehow duplicate itself or it has to be double-stranded, is what I'm trying to say. Because the retrovirus enters the cell as a single-stranded RNA or DNA. And if that happens, then it of course has to somehow be double-stranded. And this is made possible through the reverse transcriptase enzyme that will allow reverse transcription. So here's what happens. The first step is invasion of the cell as a positive RNA, and to integrate, it must undergo reverse transcription because DNA is double-stranded and the virus has to be double-stranded likewise. So it must encode RNA-dependent DNA polymerase. Now, you should have an understanding of what this means in terms of the RNA-dependent. So it's still RNA-dependent because, again, it's being translated from the positive RNA. So it's RNA-dependent. And the DNA polymerase will allow the replication, and this is the reverse transcriptase itself, the DNA polymerase, which is it's a reverse transcriptase. There are three main genes of the retrovirus. The first is the GAG, the POL, the ENV. The GAG is responsible for the capsid protein, the POL for the RNA-dependent DNA polymerase, and the ENV for the envelope proteins. And this is obviously reflected by the names are pretty nice, easy to memorize. So once the host cell has achieved lysogeny, meaning the provirus is created, the RNA-dependent DNA polymerase is inhibited. Will this seize viral genome activity? Now, so the first step of this question is to recognize what stage of the lysogenic cycle are we at the moment. And we have obviously been indicated that it's achieved the lysogenic cycle, which means that the viral genome is now integrated into the host cell genome. This means that let's look at it real fast. The whole function or the entire purpose of the reverse transcriptase is to make it double-stranded, make the positive RNA to a double-stranded RNA. And because it's already achieved and it's already integrated to the whole cell's DNA, there is no point of inhibiting the reverse transcriptase at this point. So it will have no impact on the process of the progress of the virus at this point. And we have double-stranded DNA viruses. The double-stranded DNA virus, right at the bat, if you notice it's double-stranded, it is one of the biggest genomes for the virus. It's very uncommon as well in a sense. So it's needed to encode it needs to encode DNA polymerase and DNTP synthase. And this is the wrong spelling, I forgot E over here. So the DNA polymerase, obviously, we know what that is for. And the DNTP is basically, it's a synthase is going to create the DNTP. So this is relatively large genome, encodes enzymes for DNTP production. So why won't double-stranded DNA viruses use host cell DNTPs? You have seen that RNAs are using up the NTPs available inside the cell, but why can't the DNA do, or the double-stranded DNA viruses do that? And the entire reason behind this is because host cell DNA produces the DNTP only when it replicates. And when it's not replicating, it doesn't produce the DNTP that the virus needs to replicate. Therefore, it's a limiting factor. And it's a major limiting factor. And to avoid this limiting factor, they have their own DNTP synthase. Why do positive/negative RNA viruses use cellular nucleotides? The whole reason they are able to use the nucleotides are because they're NTPs and not DNTPs. Deoxyribose is, remember, used just inside the DNA, and NTPs are freely available for the use of transcription. Because think about it, we have mRNA coming inside through transcription that will then go on to translate, and we have a lot of nucleotides or NTPs that are available for use for the purpose of RNA, but not for the purpose of DNA. So DNTP is only synthesized when DNA replicates, so it's not freely available. Therefore, yes, it has to be made by the virus itself. If the virus consists of an abnormally large genome, what might be a major problem? Well, in terms of the DNA, now because it's DNA, we will also have exonuclease activity. Well, not exactly. Well, excuse me. What I was trying to say is if we have a normal DNA strand, we have exonuclease activity, right? Because DNA polymerase will have the ability to make sure that there is exonuclease activity proofreading. But these viruses don't have that. And because of this, so let's look at the answer. RNA polymerase don't have nuclease activity. So large genomes, many mutations will happen, and this can seize virus activity. So this is the reason. Also, another limitation is because the capsid of the protein, or the capsid of the virus, cannot hold cannot hold a large genome as well. So some double-stranded DNA viruses can override cellular inhibition, making them cancerous. How will this benefit the virus? Well, this is right off the bat a continuous supply of DNTP and a continuous replication. And this makes sense because there's no cellular inhibition. There's an endless supply of DNTP, and the virus, remember we talked about the end goal of the virus is to produce more viruses, will be happily able to accept the DNTPs for its viral production. So double-strand DNA replication can happen if, number one, they provide all the means of replication. There has to be all the means of replication. Second, the infected cell is undergoing replication. Third, there is an override of cellular inhibition. These are three different factors or three different methods, or one of these have to be happening for DNA or double-strand RNA viruses to happen. So here's one example of a double-strand DNA virus, the Adenovirus. The Adenovirus is a type of double-strand DNA virus that is linear, has linear DNA. But the entire point that I'm trying to bring this up is because of the E1A gene. The E1A gene is required for transcription to happen. And if it doesn't have the E1A gene, transcription will be failed. Two adenoviruses infected a cell simultaneously. Virus 1 has a mutation for E1A virus. Virus 2 has a mutation for DNA polymerase gene. Will the infection be successful? Well, yes, because they are making up for one another's inabilities and to provide the necessary means for transcription to happen. Well, there you go. Well, this is the end of the classification of viruses, and hopefully you understand the giveaway really is the positive and the negative strand, and also the retroviruses and the double strand as well, just really pointing out the major differences inside them. And you don't have to memorize the adenovirus. That's just an example.